All right, we'll get going with this. Good morning, everybody. Any questions before I jump back into this? All right, so Friday, we left off almost wrapping up chapter 2. We left off with a couple of tables making some comparisons about the various types of microscopes and some examples of some of what we might be able to visualize with them. So we'll quickly scroll through this. The last bit of this chapter goes through some staining techniques. We'll talk in a little bit of detail about the gram staining procedure, which is what you'll be doing in the lab. And when we think about microbiology, it is one of the fundamental techniques that we utilize to give ourselves a hint of what we're dealing with. All right. So we'll talk about that here in just a second. So the bright field microscope, and I'm not going to go through and spout off everything, but it's the one that we will be thinking about as we shift over to the staining techniques here in a minute. All right. And seeing some images through that microscope like we'll work with in the lab. All right. So I believe we went through and saw all of these, the different electron microscopes, and then some of these atomic force and scanning tunneling that allow us the ability to move in even deeper and see some molecules. So when it comes to microbiology, we are typically working with glass slides that we need to, I guess, ensure that we've stuck stuff to those slides, because there are typically a lot of steps in these staining procedures where a dye is applied and then it's rinsed, and then another one's applied and rinsed. And through these rinsing steps, if you don't really have your organisms kind of stuck to that glass slide, you're pretty likely to wash a lot of it right off the glass slide. All right. So there are different ways that we may go about it. And I kind of wonder why not show a Bunsen burner up in here, because that's what we're going to think about doing. All right. But that's okay. We can get through that. So there are a few different procedures that we may employ, and it also depends on what we're really thinking about. This last image over here with some formalin, thinking bigger stuff, tissue samples or something, and those may be further manipulated as we think about like slicing to look at through an electron microscope or something else. I watched, speaking electron microscope, we were working with some of that, trying to look at some virus through the electron microscope. And there was a student that was preparing other samples with a device that shaves samples. What's it called? A microtome, I believe is what it's called. And she was getting this little, it looked almost like little thin sheets of paper, but what she was really shaving were pieces of bone, and it just went off. And with a paintbrush, she would remove it. And if it was a nice, good little rectangular shape, because sometimes they were a little bit wonky, if it was a good shape, with her paintbrush, she could take that and gently lay it right down onto a slide. So truly kind of depends on what we're thinking about. This first bit is like a hot plate. This is a slide warmer that basically, after you've prepared your slide and put whatever on there, if we're thinking bacteria, we'll spread it around on the plate, either out of a broth suspension, few loopholes onto the slide, or off of solid media like a Petri dish, you get a little bit of bacteria on your loop. And with a drop of water that was also on the loop in separate steps, you're going to mix that bacteria together in the water, kind of like the broth suspension. So you've got some liquid to smear it all around. We let the bacteria suspension dry on that slide. We say air dry, and it takes a minute or two for it to dry, and it kind of looks a little bit cloudy, right? But just simply drying it by air isn't sufficient to make that stuff really stick. We need to fix it on there, and we call it heat fixing. So using some sort of heat to get it to adhere. Technically, we can do a little bit of different stuff as well, but heat is usually what we do. So the hot plate heats that slide up, or the slide warmer heats it up and gets that bacteria to kind of glue down onto the slide. This is not a Bunsen burner, but it's like a Bunsen burner. This is an incinerator where inside of it is super hot. And we would think about typically putting like our inoculating loop inside of that, and it would cook off any bacteria that's on there. And it also helps, as it's in this chamber, helps to prevent really thinking pathogens, things that you would want to be more cautious with that potentially could be aerosolized and maybe cause some infection. It keeps it all contained in this chamber, right? It's sort of like a toaster twisted up around itself, because there's a big heating coil all the way around it. It gets nice and kind of cherry hot, and your loop goes in there and cooks off. So there'd be a lot of heat coming out of this, and that's what they're doing there with some forceps. They're just holding it right in front of the opening, where, you know, it's like opening up the oven door. You can feel a blast of heat coming out of there. We typically just use the Bunsen burner, and either with a clothespin on the end of the slide to pass it through there, or your fingertips. I prefer my fingertips with that. So as I pass it through, I can feel the heat on the tips of my fingers, but I don't want to burn myself. All right. If I pass it through once, I don't really feel anything. And that's really probably not sufficient to get the bacteria to adhere. 3 quick passes through the flame is usually sufficient to feel a little bit of heat on your fingertips, and really to know that the bacteria is stuck onto the slide. All right. With that said, with some real vigorous rinsing, squirting water right onto where your smear was at, it's likely you're going to blow a handful of those cells off of there, and you're not going to get as good of a stain as you really want. So you still are cautious, all right? But you're more likely to have it all good and stuff on there, all right? Trust me, when we get to the staining stuff, it will be all fine and good, all right? So there's other potentials, like I said, with some fixative, where we may do that with other specimens, all right? So like I said, these images are really different things, but utilizing the compound or the bright field microscopy to see some of these things. Some of it, we will think about the potentials that, to visualize in the lab, and definitely some of these bacteria we might think about working within the lab as well, all right? Not anthrax, all right? Bacillus anthracis, a true pathogen, well, we don't get to play with, all right? And I think thankfully so, all right? We don't need that, all right? But Bacillus megaterium, definitely a potential. There are a couple of other Bacillus species that we will often visualize in there as well. When we look at them under the microscope, although this is a different staining technique, they look pretty similar. Large, what we call gram-positive cells. That's what we're looking at here with the gram staining technique, although we're not giving you all of those details yet. Crystal violet is the first stain in a series of steps in the gram staining procedure. There are other potentials where maybe we might think about just a simple stain, which would be the application of just one dye and then rinsing it off and visualizing it under the microscope. And so crystal violet might be used as something like that. But you want to be real careful when you're doing something like that, because you might confuse something as a positive when it's actually a gram-negative if you use this purple dye, right? And you don't know that you only did a simple stain and not a gram stain, right? Or someone else looking at that slide doesn't know, right? So we'll touch on a little of that, and you'll see that in the lab, right? So these just some different types, really different types of staining techniques. So with this, our bacillus, the crystal violet is a basic positive stain, and that wants to stick to those cells, versus our negative stain that really kind of like a magnet. It's repelled by that, and so it's the background, not the cell itself, that's picking up the stain, right? Kind of making it seem almost like the dark filled image, but it's not a dark filled image, right? And then this one, another positive acidic stain, basic or acidic, but if they're positives, they want to adhere to those cells, they stick to them. And if it's negative, it's the opposite, right? And is repelled. So this is the gram staining procedure. There are lots of lab manuals that describe these procedures. And if you were to have a handful of the manuals all opened up to the gram staining procedure, there's going to be definite similarities with it, but there might be some subtle variations, right? The things you're adding, they're all the same, but some of the timing with it might be a little bit different, right? It will tell you in the manual that we use, these steps, basically a minute with the staining steps. And then it says a few seconds, I don't remember the exact amount, with the alcohol decolorizer step. That's the one that, you know, if you're really trying to be cautious and look at the time, that's one that you shouldn't be looking at the time. You should be looking at the slide, all right? And we'll talk about that in 1 second, so it makes a little more sense, right? So as we look at this, and we've got gram positive cocci and gram negative rods at the end, that's really what we've got, that we're showing some examples of, all right? And initially, we made our smear, we air dried it, we heat fixed it, and then we start these staining procedures, all right? So the first thing we do is add some crystal violet. We call it the primary stain. And at this point, it doesn't matter the type of bacteria, everything looks purple. The crystal violet gets on there and it stains, it's attracted to those cells and stains them, all right? After that, there's a quick rinsing step, and this crystal violet goes on for about a minute, all right? We give it a quick rinse with some water. There's some squirt bottles that allows, hold on the slide kind of at an angle, allows it to rinse off that stain. And then we add our next stain, and this is some iodine, oftentimes called Graham's iodine. The iodine goes onto these cells on the slide, and it interacts with the crystal violet, all right? We call it a mordant, and basically what it does is it helps to kind of anchor that stain into the cell wall of the bacteria, makes it a bigger molecule interacting with that crystal violet, and it gets stuck in there, all right? Especially in gram-positive cells, all right? That's really kind of the key, is it makes this big complex in a super thick cell wall layered bacteria, all right? So, but still, at this point, both types of cells look purple, look gram-positive, if you will, all right? And it's just because of the steps in this procedure. After the iodine has sat on the surface of the cell, again for about a minute, we tilt the slide, we squirt a little water up above where the smear's at, let the stain run off. And then while the slide is still tilted, we take some alcohol, Graham's alcohol or 95% alcohol, and we squirt it right up above where the smear was at so that the alcohol runs down through the smear and off the bottom of the slide, all right? This is where I said you're not looking at the time, all right? It might take a couple of seconds. It might take 10 seconds. It really kind of depends on how thick the smear was that you made on the slide. How much bacteria did you slather all around on it, all right? If it's really thick, it's probably going to take longer to decolorize than a thinner smear, right? So I look at the slide, and I'm looking at the bottom of the slide as I'm squirting water up above the smear, and I see color come off. Initially, I see some purplish kind of going to blue, light blue, and then very faint, and it's colorless, right? As soon as it goes colorless, I have decolorized my slide. And like I said, time doesn't matter, all right? Couple of seconds, all right? And it stops giving me any sort of hint of color. And then I squirt water up above that smear to stop the alcohol decolorizing process, right? You can use not enough alcohol, and you can use too much alcohol, right? And it can definitely mess up your gram stain, all right? Not enough alcohol can make gram negatives look gram positive. And too much alcohol can make gram positives look gram negative, right? But you oftentimes see some hints of the right stuff on the slide, all right? So you're like, wait a minute, right? What did I do, right? I'm getting some purples and pinks together, and they all look like the same sorts of cells, right? You get purples and pinks that are different looking, rods and cocci, well, you've got different stuff going on. But if it's all really the same looking stuff, maybe it's a little bit of your technique, all right? It takes some practice to really kind of get a feel for it, all right? So after the alcohol step, and we've rinsed it with water, if we were to put this under the microscope, our gram-positive cells look positive, they look purple, but our gram-negatives look hollow. They look kind of like ghost cells. You can't really see them. Potentially the hint of a cell wall is visible, but it's difficult, right? Especially if you've never really looked at much bacteria under the microscope, you're going to be hard pressed to find it, all right? So the alcohol, when it gets added to the slide, if it's a gram positive, they've got that super thick peptidoglycan layer, and that iodine crystal violet complex is stuck in there. And the alcohol doesn't really do anything to it, right? But the gram negative cell that's got a very thin peptidoglycan layer, single layer thick really, and the outer layer, that's really the kicker. The outer layer that's a liquid, or not liquid, a lipopolysaccharide, a fat sugar layer that is an outer membrane. When the alcohol touches that, it causes the lipid layer to dissolve. And now that little thin layer of peptidoglycan, the alcohol hits it, and only a single layer thick, it just rinses right off, right? And the purple drips right off the end of the slide, all right? And so that makes those cells look colorless. And in order to visualize both of them, we do a counter stain. Safranin is a pink or reddish colored stain. And when it's added to the slide, gram positives that are already purple, you add some pink to purple, you get purple, right? It doesn't do anything to them, right? But gram negatives that were colorless, now they pick up that pink stain, and now we can see them. So it allows us to differentiate. It's a differential stain, letting us see gram positives, thick cell-walled bacteria, and gram negatives, thin cell-walled bacteria, right? And we get all sorts of details. We can see the shape, rods or cocci. We can get some morphology, chains or clusters. Bacteria that are rod shaped sometimes are linked together end to end, and we call that porting. It's not something that's as diagnostic when we think about looking at a gram stain compared to some of the cocci when we see chains or clusters, because typically I see chains, and we call that a streptococci are long chains of bacteria, and a chain of them like that might make me think about strep throat, Streptococcus pyogenes. Clusters of gram-positive cocci makes me think some staphylococci, and staph is clustered, and that might make me think of maybe a staph infection, Staphylococcus aureus, as a potential. All right. But simply seeing a chain or a cluster of them, I really can't say 100% that that's what I'm dealing with. There's more things that we will do, all right? But we've got some hints, right? And truly, it may dictate the types of media we might try to grow them up on, and it also might give us some hints about the types of antibiotics that we might utilize to treat some of these. Some antibiotics are more specific toward gram-positives, others toward negatives, and then there's some broad spectrum antibiotics that kind of don't care. It's just more of a kill them all approach. They target all sorts of types of bacteria, negatives and positives similarly. So here is an image, a little bit fuzzy, but an image of a mixture of bacteria, which oftentimes in a clinical situation, that's kind of what we're going to be dealing with, is a handful of different things together. So here you can see some cocci, purplish cells, and they look clustered, grouped up together, all right? Here's a pretty good image of some clustering, all right? There's a few that look sort of just paired up. We might call that diplococci, but when you get the bigger picture of it all, it looks to me more clustered than anything else. The more sort of fuzzy stuff, the backgroundish looking stuff, sort of, this is more pink. And if we kind of had it zoomed in a little bit different or focused a little bit better, because I feel like this isn't the best image. These are little gram-negative cells, all right? And E. coli or Escherichia coli, E. coli is teeny on the microscope slide, very, very small. Bacteria are small in the first place, and E. coli is little. And oftentimes looking at it, it almost looks sort of like a little pinpoint. And many times you look at it and you're like, I think that's a gram-negative cocci. It looks like a little circle, all right? But it's really not, all right? And sometimes you might see a little bit of both, all right? Is this pen gonna work for me? So a little dot like that, that's not the best dot because I wiggled my hand. Or maybe you see a little more rod shaped, all right? You might see a bit of both of these things going on, especially maybe just thinking E. coli on this slide, not a mixture, all right? And you're like, oh, there's little dots and then there's kind of a little bit bigger rods, right? I've got rods and cocci on there. But you got to think about it a little bit, flip it around from you to the bacteria. These suckers are super little, all right? And if we lay them out onto the slide, maybe part of you thinks all of these cells that are rod shaped should just lay down flat, but they don't. Some of them are standing up on their ends. So you might be seeing the top bit of it, or you might be seeing the side of it, all right? If they're cocci shaped, they're just little balls and they're all over the place. So you're not seeing it like that. But the rod shaped bacteria, some of them might be standing up on their ends, all right? And that also might make you a little bit confused as you're looking through there, right? Just kind of depends. So here's another one, just a straight pure culture, if you will, only one player. And this one's not telling us, but they are positive cocci in clusters, some sort of Staphylococcus, could be aureus, it could be Staphylococcus epidermidis. There are technically a boatload of other species of Staphylococci, but these are two of the main ones we really think about dealing with or maybe harvesting off of our bodies. And that also depends kind of on where we might try to collect them as far as the different type of staph that we might find. So shifting over, this is a specialized type of staining procedure. It allows us the opportunity to see things that really would maybe be lost on a gram stain, all right? There are some bacteria that have a, not like a gram negative with that lipopolysaccharide layer, but a different substance that they secrete outside of their cell wall that gives them some protection. So mycobacteria, like our tuberculosis here, and there are a handful of others, and some other genera, but mycobacteria is the main one we think about in here. These bacteria are what we call acid fast, and they secrete a substance outside called mycolic acid. And it's basically like a wax layer, a waterproofing mechanism is kind of what I think about. And it makes it almost impossible for the gram staining reagents to penetrate through that wax layer. And you oftentimes don't see anything on a gram stain, all right? Depends kind of on the circumstances. If it's a old culture of mycobacteria, some of those cells kind of starting to die, their cell walls starting to degrade, the wax-like layer breaking down, it can pick up some gram staining reagents, right? And they would appear gram positive, all right? Technically, the mycobacteria are gram positive organisms. They have a thick peptidoglycan layer, but we don't call them gram positive really. We call them acid fast because they don't typically pick up the gram reaction, right? And that was something that I had a little bit of a struggle with just a little bit ago, because I was pretty sure these guys they're not gram positive, right? But technically they really are. They've got the thick peptidoglycan like the other gram positives, but it's the extra mycolic acid that sets them apart. So here's the situation. You've got an individual that has some sort of chest symptoms going on, pneumonia-like symptoms, something, right? Some sort of issue. And they present to the clinic. You collect a sputum sample, we stain it up. Gram staining gives us nothing, right? But we're thinking this person's truly sick. We're thinking some sort of bacterial culprit. And in turn, visualizing nothing with the gram stain raises some alarms. And we do our specialized stain. The acid fast procedure allows the stain to penetrate through that wax layer. And now it shows up as these long, pink, filamentous looking bacteria, big threads of bacteria, all right? This is a whole glob of them together, all right? Many of them. And the blue stuff in the background might be just the counter stain. It also can show us other bacteria that also might be present, but not acid fast. And they pick up the blue dye, all right? So this is the only staining procedure that we can look at in the lab and immediately diagnose that patient. Like I said, with the gram stain, you can't pinpoint it and say, but this, if we know this came from the sputum of a patient and we see the acid fast bacillus, you've got tuberculosis. It's a done deal. And we start treating with antibiotics immediately, right? There are all sorts of issues with drug resistance and other problems. We still want to cultivate it. We want to grow it up in the lab, but it takes a long time to grow. Most bacteria, we can get cultures of them the following day. And these guys can take weeks before you get visible growth on that media. And if you didn't know that's already what you're trying to get in the first place, you'd write it off as there's nothing there to begin with. But there is, right? It just takes a long time, right? So this, an example of a specialized stain. India ink is Another negative staining procedure that stains the background of the slide, but not the cells. Here is some cryptococcus. These are some yeast cells, and really it's a good one. This isn't the best negative stain, but it can show some capsule layers around the cells. And cryptococcus, being a yeast cell, has a very thick cell wall that oftentimes makes a nice line around the circular yeast. And outside of it is a sugar coating, a capsule layer that gives it sort of a halo looking. So it's sort of two-toned, dark line. I mean, it's still all hollow in there, but dark line around it. And then the outer bit, that's the halo. This isn't the best really to see a capsule, but it says it's got them. All right. All right. Next door. Here we are also seeing a different type of stain, right? And this is another procedure where we're looking at some bacillus. And in this case, we're really trying to see some spores, although this says encapsulated, seeing that, but you can also, and the spore stain's coming up, but you can see some in these cells. But this is really trying to show us kind of like our negative staining, a capsule layer, a sugary coating, right? Lots of bacteria have some capsule layers that they produce on the outside. Not all of them, but a good chunk of them do. It gives them some protection, gives them protection outside in the environment, gives them some protection inside of a host to potentially go undetected by our immune response, right? We'll touch on some of that kind of repeatedly as we move through here and talk about some of these mechanisms that cells may have. So this, was that Bacillus subtilis before? Just says Bacillus cells. So this Bacillus subtilis is definitely a bacteria you guys will see in the lab. These are gram-positive cells, purple colored, but this isn't a gram stain that we're looking at, right? So another one that you might initially look at and be like, oh, we use saffron in here. These must be negatives, but you didn't do a gram staining procedure, right? You use saffron in as just a simple stain, just as a dye, not something to tell us, is it gram positive or negative, all right? So really what we're looking at is not the pink cells, it's the green stuff inside, all right? So some bacteria, and for our purposes in here, we want to think Bacillus and Clostridium. Those are the two genera of bacteria that are spore formers, right? And one extra tidbit, Bacillus is a gram, well, they're both gram positives. Bacillus is an aerobic bacteria, likes oxygen, and Clostridium is an anaerobe, doesn't like it, all right? But they would both appear gram positive under a gram stain. And sometimes when we gram stain these bacteria up, we may see some hints, little hollow bits inside of them that might make us think maybe that's a spore, right? Spores are protective mechanisms for these two genera of bacteria. It allows them to survive almost anything, all right? Lots of adverse conditions, lacking nutrients, altered conditions, too hot, too cold, the pH shifts, whatever it might be. Things that the cell may recognize as, holy cow, this is going to kill me, makes it stimulate this spore formation, a protective structure that allows it to go on and live another day later on, right? So endospore staining techniques allow us to determine if the cell actually has the capabilities of doing this, or potentially we may be seeing something inside of that bacterium that just didn't pick up stain, like maybe an inclusion body, something like foodstuffs that have been pulled in and not broken down. We may pick that up, and we need to differentiate that, all right? So the spore formation is a protection. We go through like we're going to replicate. We copy our DNA, but instead of replicating and creating 2 new cells, we take that replicated DNA, package it up with some enzymes, and coat it with numerous coatings to protect it, like I said, from almost everything, right? And oftentimes we'll get some free-floating spores if we've got an older culture and you don't see the vegetative cells. This is still lots of cells still forming spores, right? That's the idea. So this next image is showing us the flagella of bacteria. Some bacteria have flagella and some don't. If they have it, we're thinking some sort of rod-shaped bacteria. I can't think of a coccus bacteria that's got flagella. I don't think they do, right? So rod-shaped could be gram-positive, could be gram-negative, right? And then as far as the way the flagella go, there are all sorts of variables. It might be a single flagellum. It might be one on either end. It could be a tuft at one end, or it could be like this one right here, all over, completely covered, like hair all over the place, all right? And there's different names for all of that. We're not going to go through that right now, all right? So Bacillus cereus, another bacillus, one we'll look at in the lab as well. This one is a potential culprit for some foodborne illnesses. And here we can see the flagella with what we call a flagellar stain. This is a kind of labor-intensive procedure. It takes lots and lots of dye applications to get to the point that we've coated the flagella enough to make them visible under the light microscope. Usually you just can't see them. They're too small, all right? And this is a procedure that we just don't deal with in the lab. It's too difficult to really make it work well. All right, so wrapping this up, basic or acidic stains that are positively charged, or this one, it could be negative, depends on down below, but allowing them to stick, and then the negative stains that are repelled by that, by the cell wall, and in turn giving us some different images. So gram stains, like I said, the main one we think about in microbiology, and then some specialized stains. Capsule I'd put up above the flagellar stain because it might be something you'd think about more than others. So here's an ultra microtome. I told you about the student that was slicing up the bone. This one allowing to shave with a very super sharp blade on here. As you twist it, it gives you these little slivers of whatever it is you're trying to get. And that's what we're getting over here, same sort of an idea to visualize. So we showed some of these spirochetes, this tail end of this bit. So with a dark field microscope image, we can see those spirochetes. We can use some silver staining, a procedure that we didn't show as one of these steps, but similar sort of an idea where they're attracted to these cells, giving us this image, and then some electron microscopy to visualize the little corkscrew cells. With that, we can also think about using some fluorescence techniques with our treponema cell. So antibodies can link up to it, and then a secondary antibody that's got the fluorophore tagged to it is attracted to that antibody. They link up, and then with the fluorescence microscopy, it glows. So giving us the opportunity to visualize them. I thought I was going to get the glow in the dark one to see those. All right, let me stop this share. Let me just introduce this next bit. Maybe. All right, questions about any of that? Staining. What? Where did you go? I don't know what I did there to stop the share. Okay, so shifting over to cells, we want to think a little bit about details of eukaryotic cells and prokaryotic cells to kind of get some ideas about what's going on, right? I hope the majority of this chapter is something that is review as we move through here. It really kind of depends on, were you in biology or did you take 2227 as far as some of the details that we may have covered or glossed over as we get in here. So I hope a lot of this is sort of review-like, all right? There are all sorts of different potentials as we think about microbes from, eukaryotic cells with a lot of business going on their insides to our prokaryotes that are pretty bare bones, where we might expect to find them, all sorts of variation when we think about it. And with that, I think, I'm not going to go off on all of these details, but some really pretty impressive cells when we really think about it. So as we start this stuff, we need to think a little bit of history about this and how we come to our understanding of some microbiology in the first place, all right? There were all sorts of experiments, and we talked about Louis Pasteur and his pasteurized milk. We'll talk about him again soon. There were all sorts of thoughts about how things worked, and we recognize now that these thoughts really didn't make much sense at all, right? How did it get there? Just from the abyss. Spontaneous generation, right? Foul smelling air, something like that, right? That potentially caused whatever it is that we're observing, right? And some started to question that a little bit and think maybe there's another reason, right? Maybe there's something else to it, right? And set out to establish a series of experiments to really kind of show that they were on the right track, right? And with that, it's not something that happened overnight, right? When you've got a specific belief mechanism, a belief system in place, to get someone to change their mind is pretty stinking difficult, right? People are stubborn, right? This is what I've always known, and that's the way it always will be, and you can't change my mind, right? It takes a long time to truly get people to change their minds, right? Super long time, like potentially centuries, right? Spontaneous generation took a super long time to finally drill that final nail in the coffin, right? Although we move through here and look at this and think, oh, well, that seems pretty straightforward, all right? We're already coming in here biased, having some ideas about this to begin with, right? It didn't happen overnight, right? So there's lots of things that I start thinking about that, maybe we take for granted. We go to the supermarket, we come home, we put all of these perishable things in the refrigerator, right? It stays good for quite a while, right? There could be some Tupperware pushed to the back of the fridge that has some crazy stuff growing in there, right? You're like, what? What was that, right? It doesn't look anything like it used to, I'm sure, right? But back in the day, refrigeration, well, all of these things unheard of, right? So the potential for things to spoil is huge, right? and keeping things good for time frames like we think about just is unheard of, right? So meat, as we see here, the potential for it to not be so good, probably pretty common, right? We could think about all sorts of other things like, water, right? Very unclean. You're not really drinking that stuff. Not if you want to stay healthy. You're gonna have to consume some beer, right? That's what lots of, you want to drink something, you're gonna ferment it, right? Get a little alcohol in there. Now it's better to, better for you, right? Is it truly? That's beside the point, right? So here we've got a series of experiments, really a series, because it wasn't just like this, all right? You might recognize meat that's been out for a bit of time to all of the sudden have some creepy crawlies all over it, some maggots that seem to be feeding on your dinner, right? How did it get there? Where did these things come from? Spontaneous generation, right? It must be. They just showed up, right? We know that fly has laid some eggs on the meat and they hatched out, and these maggots are feeding on it, right? And in turn will emerge as some flies, right? Or at least I hope we know that, right? So Francisco Reedy set out this experiment to show that these maggots really are the result of these flies. It's not spontaneous generation, right? And so, like I said, really it's a couple of experiments. The first one was the open flask and the cork, where no flies could get in because it was corked up, right? And in turn, the open one has maggots, the corked one didn't, right? Done deal. Seems to show, but like I said, it's hard to get everybody on board, right? There's lots of issues, right? And the naysayers are like, it's because you corked it, right? You need to have air in there. If you don't give it any air, right, which is where all of this must be coming from in the first place, if it doesn't have contact with the air, that's why, right? So he sets up another experiment. And in this time, puts some gauze or some cheesecloth over the top of it so air can get in, but he keeps the flies out, right? And in turn, the meat doesn't get maggots. The maggots ultimately are up in the cheesecloth. They smell the nasty meat down there, but they can't get into it, right? Another one that we're like, done deal. But we're not, all right? It seems so simple, right? Seems like that's enough, right? Maybe with bigger things, maybe with these maggots, multicellular sorts of stuff, maybe that makes sense. But careful observation of this meat that we're not showing you here, maybe thankfully, right? This meat's got all sorts of other stuff going on. Gets a little bit slimy, putrid, right? There's some bacteria that are feeding on all of this stuff. You can't see them, but you can kind of see some of the ill effects. And I bet you can sure smell it, right? It only takes a little bit of time of that meat being on the bench top at room temperature before it starts smelling real bad, right? And let it sit there for 5 or 6 days. I bet you'd prefer to be outside, right? Where'd all that stuff come from? That's where we're at with the next bit. And we'll pick up with that on Wednesday. All right. Farewell. Peace be upon you.